Hello, my name is Mike Bullivant, and I'm here to introduce this particular video sequence. We're about to take an insight into the world of antibiotic research, and we'll be following the development of two antibiotics along very different routes. All the chemistry involved is still as correct as it was when this video was first presented 10 years ago in 1989. But like most areas of science, antibiotic research has come a long way since then. To give you an insight into some of the advances, I'll be appearing at various points in the video talking to Dr. Mike Marriott, Therapeutic Director of Microbial Diseases at Glaxo Wellcome. Mike, thanks very much for coming along. Now, the video we're about to see concentrates on the beta-lactam antibiotics. Now, how significant a discovery were these?
Well, these two particular compounds, when they first appeared in the late 1980s, were very significant discoveries indeed. Well, they still are very significant discoveries, obviously. They represent probably the pinnacle of activity for their particular class, and really we felt that it was going to be impossible to improve on that activity.
So you thought that they were going to solve all of the
We were convinced that they would solve all the problems of the world, yes, that's true.
Yes, but as I said in the introduction, I mean, it has come a long way since then.
Oh, indeed, yes. And, well, as it happens, it turned out we were wrong, because the bacteria are awfully clever little individuals, and they have managed to become resistant to both these and just about every antibiotic that we have.
Yeah. So there have been some significant advances over the last 10 years.
Oh, there have been some enormous technological advances in the last 10 years. And watching the video, I'm just struck by what progress we've made and what an impact these new technologies are having on the whole discovery process.
Right, well, thanks, Mike. We'll be discussing that later. Well, now for the video.
Take a mixture of carbohydrates, fats, proteins, vitamins and minerals, add a gelling agent, warm to about 50 degrees C, pour into a large Petri dish and allow the mixture, called agar, to cool and set. If it is then removed from the sterile area that it was prepared in, it will become contaminated by airborne fungi and bacteria, which, in the nutrient-rich medium, grow to form colonies. Close inspection of these colonies shows that they don't all grow freely. Some microbes have the ability to inhibit the growth of other colonies.
It was just such an observation in 1929 by Fleming that led to the discovery of the biocidal nature of Penicillium species. This led to the birth of the antibiotics industry, and this industry has now grown to the astonishing extent that last year the turnover was £9 billion. The most important of the antibiotics are the beta-lactam antibiotics. These are the penicillins, the cephalosporins, and the phenomycins. We're going to consider two case studies. We're going to look at two very important drugs which have been discovered in recent years. But this raises a question. If the industry has been so successful over the past 20 or 30 years, why do the companies continue to throw so much effort and so much money at the problem?
The explanation starts here. This is cephalosporin C, the first compound in one of the most important classes of beta-lactams, the cephalosporins. It occurs in nature and was originally isolated from a fungus found growing in a sewage outlet in Sardinia in 1947. It's the lead compound from which a whole family of antibiotics has been made, but it isn't itself a particularly powerful antibiotic. Why? It all hinges on the way that antibiotics work. What they do is disrupt the process of cell wall biosynthesis. In a growing colony, this process is taking place all the time, both to maintain the existing cell wall and to make new wall as the bacteria divide. When antibiotic is added to a colony, this inhibitory action has two effects. Firstly, the bacteria are prevented from successfully dividing, so they start to form long, spaghetti-like strings. The other effect arises from the fact that the existing cell walls routinely break down and are in constant need of repair. As the process of repair is prevented, small flaws appear, large enough to allow water to start moving into the cell due to the high internal osmotic pressure. As a result, the cells start to swell and eventually burst, a process called lysis. However, not all microbes are equally susceptible to antibiotic attack. Two main factors influence this. The first is the nature of the cell wall. The early antibiotics, the penicillins, were only successful against bacteria with a relatively simple cell wall, the so-called gram-positive bacteria, which turn purple when treated with the gram stain. The gram-negative bacteria, which turn pink under the same conditions, have a more complex cell wall, which acts as a barrier against antibiotics, and so they are not so readily attacked. The cephalosporins had a special feature which allowed them to penetrate this barrier and attack the gram-negative pathogens. But there is a further problem. Bacteria themselves can mutate to produce enzymes called beta-lactamases, which break down antibiotics before they reach their target. So, in addition to the need to penetrate the outer barrier, the antibiotics must also have a resistance to attack from these beta-lactamases. So cephalosporin C, while it pointed the way for future drug development, was neither very resistant to beta-lactamases, nor was it successful at attacking a broad enough range of bacteria. Since its discovery, the drive has been to produce antibiotics which will attack gram-positive and gram-negative pathogenic bacteria. How successful has the pharmaceutical industry been at achieving this? A simple demonstration illustrates the point. The vast majority of infections are caused by only 11 families of bacteria. Pure cultures of 8 of the most common of these pathogens are smeared onto a plate of agar in long, straight lines. These inoculations will take nutrients from the agar and will develop into large colonies on incubation. But before this is allowed to happen, strips of paper impregnated with different antibiotics are laid across the inoculated lines. As the bacterial colonies grow, the antibiotic will diffuse out into the agar, coming in contact with the bacteria. After a couple of days, the bacterial cultures appear as dull, creamy lines, but they are not complete lines. Many are broken up where they cross zones of agar into which the antibiotic has moved from the paper. The degree of inhibition shows the success in increasing the spectrum of activity over the years. The early antibiotics were only successful against a couple of pathogens, while the later ones have attacked an increasingly broad range of them. Before the advent of the two drugs we're going to look at, there was one major target left, a bacterium called Pseudomonas aeruginosa, which had proved to be a particularly difficult pathogen to attack. This was what the chemists were aiming at. But this in turn begs the question, why are chemists looking at microorganisms? Has the chemist turned into a biologist? The classical image of chemistry is that of the alchemist offering his latest elixir, which he has just synthesized to a hapless patient. How far is that from what the pharmaceutical chemist does today?
The chemist really has two roles to perform. Firstly, to define what to make, i.e. to translate that biological hypothesis into a chemical hypothesis. And secondly, he must know how to make them. And it's therefore vital that we employ good synthetic organic chemists in order to be able to make those target molecules.
In other words, there's more to pharmaceutical chemistry than what's in a flask. Before any synthesis can take place, the chemist must have a clear understanding of the medical condition he's going to treat. How did Glaxo set about translating their biological understanding into a product?
Once a new project is defined, the strategy for that project, it is essential to bring together the people from the disciplines that are involved and to find somebody to lead that. And that person must be somebody who is thoroughly conversant with the whole spectrum of biology and has the qualities to get others to respond to his leadership. In this case, Chris Neal was the person who was chosen because he had a long experience working in antibacterials and was a chemist with a considerable commitment and flair.
At the beginning of the project, our objective was to produce a cephalosporin antibiotic which was stable to beta-lactamases and had a broader spectrum of activity than any of the previous antibiotics. And this had to include activity against Pseudomonas aeruginosa, a very resistant organism which is responsible for, particularly for infections in hospital with patients who are seriously ill, particularly people with severe burns and cancer. In order to explain how we set about this, the best way is to take the analogy of a military vehicle. If one regards an antibiotic as a military vehicle, and a cephalosporin in particular, a cephalosporin is typified by a 4-membered beta-lactam ring which is linked to a 6-membered ring, and this provides the main features that we're going to require. Firstly, the beta-lactam moiety provides the bullet, and this is supported by the remainder of the beta-lactam structure which holds it into the right shape. That is effectively the body of the vehicle. In our case, we need some armour against the enzymes that the bacteria is producing to try and destroy the antibiotic, and this can be provided by an oxime function here. And then finally, of course, an antibiotic is no use at all unless we can get it through the body, distributed well in the body, and into the bacterial cell that we're trying to attack. This is effectively provided by the wheels of the antibiotic, which come from the remainder of the molecule here. Now, the compound we've illustrated here is the final point that we got to at the end of our investigations. This is ceftazidime.
So the final molecule can be analysed in terms of the function of each of its components. With hindsight, it's easy to see the significance of each of these moieties, but this is the result. How did Chris Newell end up with this molecule? His starting point was this: 7-aminocephalosporanic acid, or 7-ACA for short. It was chosen because it was readily available following removal of the side chain from cephalosporin C, which Glaxo produced by fermentation. So, out of all the groups he could have permutated around the molecule, how did he approach the problem of finding the groups that he ended up with?
Ceftazidime arose as a result of many years of effort, starting from a starting point where we had molecules which already had an oxime function which was providing very good stability to the beta-lactamase enzymes, which many bacteria use to destroy antibiotics. This oxime function, more particularly a methoxime, was modified by ourselves, and we found that by modifying the methyl group of the methoxime function, we could improve the spectrum of activity, and in particular we could introduce some activity against Pseudomonas.
It was generally known that the oxime function conferred beta-lactamase stability. A logical extension to that was the addition of a methoxime function, and subsequently its replacement by a highly elaborated oxime which also had the benefit of improving the pharmacokinetics. What about the aminothiazole moiety?
In our early work, we'd found that the furan ring gave us the best overall activity in that particular methoxime series. However, now that we were modifying the methoxime, we began to re-examine the heterocycles, and after looking at many variants, we found that an aminothiazole provided a broader spectrum of activity generally right across the board.
So, because of the discovery of the activity of the oxime function, he changed the heterocycle and arrived at the aminothiazole, which was a better wheel for the military vehicle. Finally, the pyridine ring at the southeast corner of the molecule.
The pyridine ring in the molecule again came from a wide range of variants, and amongst these we found that pyridine gave us the best overall pharmacokinetics and the best metabolic stability of all of them.
And hence the final product. But a word of caution. In taking the molecule apart and discussing the different functional groups in this additive way, there is a suggestion that the result was logically derived from working on each of the separate moieties, finding the one which showed the greatest activity and moving on to the next in a piecemeal way. This isn't the case.
During this program, we tried many hundreds of variations on the structural features that I've described. Of course, things are not quite as simple as they seem, otherwise we would have got to the compound straight off without really doing any research. In drug discovery, we find that properties are not always additive, and that although a structural feature may provide an advantage in one particular direction, its effects on the rest of the molecule in terms of biological activity can be quite disastrous. In this game, 2 and 2 is very rarely adds up to 4. Most often the answer is 1 or 2 or a complete lemon. However, on very occasional molecules, you get a synergy of activity between the various parts, and 2 and 2 can add up to 15 or even 20. In those occasions, you've hit the jackpot, and you've really got something to go for.
Well, we saw there how that particular antibiotic, ceftazidime, was arrived at. Now, Mike, Chris Newell, who we saw in the video, made it clear that it isn't just a case of combining groups which have the required properties and hey presto. It's more a matter of playing around with these moieties until you get the right combination, you know, the complementary combination. Now, that would seem to me to be a very time-consuming process, is it?
Oh, that is a very time-consuming process, exactly. Far too time-consuming today. We have to change it, we have to use new technologies which will allow us to make many more compounds in parallel. This is the technology which we call combinatorial chemistry.
Now, what is combinatorial chemistry, just briefly?
Well, if you can imagine that it arose from solid-state peptide synthesis, take the 20 naturally occurring amino acids and combine them in threes as tripeptides, all the possible sequences would be a library of about 8,000 compounds. If now you go and look at all the unnatural amino acids and combine them in as well, you can create libraries of 50 or even 100,000 compounds.
Now, with the compounds that you're talking about and researching, I mean, what kind of scale are we talking about there?
Well, we have a small biotech company in California which builds a machine about the size of these bookshelves. And that machine is capable of making roughly 48,000 compounds pretty well overnight.
Overnight?
Yes, indeed. There's a little bit of an exaggeration. There is some work that has to be done in advance to get the conditions for the reactions right. That might take a few days, it may take a few weeks. But once that's done, all the scientist has to do is to load up the reservoirs, flick the switch, and in the next 24 hours, all 48,000 compounds will be made.
Quite astonishing. But we're not talking about synthetically pure compounds here, surely?
Oh, yes. Each compound is synthesized on a bead, on a plastic bead. And each bead is tagged so that you know the order in which the molecule was put together, so that we can go back and remake the compound if it turns out to be active on subsequent screening.
So when you've got this vast array of compounds, I mean, what do you screen them for?
We have in vitro screens now for looking at things like solubility, certainly, whether the compound is likely to be orally absorbed, whether the compound is likely to be cytotoxic, whether it's likely to be metabolized. And all that information is stored on the database along with the activity of the compound as well.
I understand that Glaxo welcome that you have a hit-to-lead policy. Now, could you just describe what is a hit and what is a lead? What is that policy?
Well, a hit is a compound which is active in one particular screen. A lead is a molecule which has most of the properties that you believe you require in the final drug. It just needs a little bit of finishing, a little bit of polishing to convert it into the final drug.
So you've got far fewer leads than you have hits.
Exactly, yes.
I mean, what's the ratio?
Oh, the ratio I wouldn't like to say. It's probably 100 to 1 or something like that. It meant that back in 1989, when we had very few hits, we were much less discriminatory. We would accept almost anything as a lead. Now we're much more demanding. Our policy is to have most of the properties we require in the final drug before we're prepared to call a compound a lead.
So that hits-to-lead policy has changed significantly over the last 10 years.
Exactly.
Yeah. Well, after the problems of research to obtain a very active antibiotic, what about the synthesis of it? And how was ceftazidime put together? Chris Newell.
When we're doing synthesis in a research lab, our main objective is to try and get to the compounds we want for evaluation as quickly as possible. And we don't really take into consideration the yield or the synthetic efficiency or the cost of the starting materials. By and large, we grab any intermediates that are convenient and convert them into the final product that we want to evaluate. However, once a compound has been identified for serious development, we obviously have to reconsider the synthesis, because the final synthesis for production must be cheap, it must be economic, That is to say, it must work from a cheap and economic starting material. It must be extremely efficient. It must be easy to undertake in a factory environment. And above all, it must be safe. In the event, ceftazidime is made from cephalosporin C, a fermentation product that has been produced by Glaxo for over 30 years.
To start with, cephalosporin C is converted to 7-ACA, with the carboxylic acid protected as the tertiary butyl ester. In a convergent route, the aminothiazole had been separately synthesized, with the amine and carboxylic acid protected. This is reacted using a DCC coupling with the amine of 7-ACA in a single stage. Following the completion of this reaction, the protecting groups are removed by trifluoroacetic acid. The final stage is the replacement of the acetate by the pyridinium group by simply reacting with pyridine in the presence of sodium iodide. A straightforward and concise transformation from a readily available intermediate.
Glaxo has a philosophy that we should try and keep our research as simple as it possibly can be. And in this case, using 7-ACA as a starting material, I think we've demonstrated that by using synthetic chemistry that is not all that complex, we've been able to derive a molecule which has considerable benefit to the patient.
Ceftazidime as a drug is derived directly from a program of what can be described as rational semisynthesis. It is in fact the most important member of the third generation in a line of cephalosporins which have been developed in this manner.
Well, that concludes our first story of antibiotic development, tracing the synthesis of ceftazidime. Mike, we finished there with the third generation of cephalosporin beta-lactams. Now, just how successful were these drugs, and is there any residual work still being carried out on them?
Well, ceftazidime has been a very successful compound, a very successful drug. There has been a little bit of work that's gone on subsequent to ceftazidime, and one or two compounds have been marketed which are sometimes referred to as fourth generation cephalosporins, but they don't really offer many significant improvements over ceftazidime, and really work in that area has more or less stopped. It was very difficult to improve on the activity of ceftazidime. We found everyone else in the industry who tried found the same. Worse than that, as we made the molecule ever bigger, trying to find new ways of varying the groups around the periphery of the beta-lactam nucleus, then we found we were introducing toxicity into the molecule as well, which was completely undesirable.
So tweaking the group actually was counterproductive?
Exactly, yes,
So
ultimately.
Is it fair to say that beta-lactam research has died a death then?
Cephalosporin research, yes. But we still have some work going on in the beta-lactam field. We discovered a novel series of tricyclic carbapenems, so-called trinems, in Verona about 6 or 7 years ago, and we're still carrying on work there.
How do they differ from the cephalosporins?
Well, they, rather than being a 4-6 bicyclic molecule, the beta-lactam is a 4-5-6 tricyclic molecule.
Much more complex.
More complex, yes. They are fully synthetic molecules. They're not made by semisynthetic synthesis in the way that ceftazidime is.
And you're still taking these molecules and tweaking them?
Oh yes, we are, definitely. We believe that they do offer advantages over the cephalosporins in that they are more stable to beta-lactamase enzymes. These are enzymes that the bacteria use to degrade beta-lactams and make them resistant to this class of antibiotics.
So, since beta-lactam research finished, has anything else sprung up from the work? Any recent developments?
Beyond the trinems, no. There hasn't been any progress within the beta-lactam series. There's been a lot of effort going on in other classes of antibiotic, and a lot more tweaking going on in other series of compounds, certainly.
Yeah, so it really is time to move on to newer compounds?
It's time to move on, yes. I mean, in every series of compounds, I think we've got as far as we can go easily with the currently existing antibiotics.
The synthetic approach is not the only way to discover antibiotics. An important tool for the pharmaceutical chemist who's looking for microorganisms that produce antibiotics is, believe it or not, a shovel.
The soil is in fact an extremely logical place to look for new bacteria and fungi. It's a fantastically rich microcosm, every gram containing tens of millions of microorganisms. 70% of these are producing antibiotics, probably as a self-defense mechanism. It's the original germ warfare. Any one of these microorganisms may be routinely producing an extremely broad-range antibiotic which could be medically useful. Selected bacteria and fungi are carefully isolated and cultured until there's sufficient material in which to carry out an evaluation or screen, as it's known. The quickest and most visible way of screening for antibiotic activity is to isolate a sample of the growing microorganism. In this case, a fungus with an extremely striking colour imparted by the sporing bodies. And then place it on an agar which has been contaminated with the pathogen we're trying to kill. A couple of days later, the clear halo around the fungus is evidence of antibiotic activity. But the screen which threw up our second antibiotic was more sophisticated than this.
Approximately 20 years ago, Merck embarked on a radical change of course. Rather than using the traditional screening for antibiotic substances, we employed a specific mode of action type screen. That is, we were looking for compounds which did one specific thing biochemically. Our choice of target was partly facilitated by the observation that penicillin was a cell wall biosynthesis inhibitor. We chose the inhibition of cell wall biosynthesis as our specific target. We like this target for two specific reasons. One, the compound, any compound that inhibited the bacterial cell wall would create a defect in the cell wall, and this would cause the compound to literally explode from the high internal osmotic pressure. It's sort of analogous to a balloon filled with water and then pricking it with a pin. The other reason that we liked this mode of action screen was due to the fact that the bacterial cell wall has no counterpart in mammalian physiology. So anything we could do to disrupt that would not be toxic to mammals and hence people.
Their screen involved the observation of bacterial cultures in the presence of soil isolates. They were looking for evidence of disruption of cell wall synthesis, the known effect of beta-lactam antibiotics. What they found with one isolate was that the culture was prevented from multiplying, with the individual bacteria slowly swelling as the integrity of the cell wall was destroyed. The final result was lysis of the bacteria.
Naturally, we were very elated when, in the early 70s, we came up with a culture which indeed exhibited all of these features that we desired. It was broad spectrum, it was a bacterial cell wall biosynthesis inhibitor, and it had very excellent anti-pseudomonal activity. What we had not built into the screen, and what we very quickly determined, was that it, in addition, it had beta-lactamase stability, which was a very, very desirable plus to the very excellent profile that this culture already exhibited.
So what was this compound? Mass spectrometry revealed its molecular weight and molecular formula. Infrared spectroscopy picked out a number of functional groups, a carboxylic acid, an amino group, a hydroxy, and most importantly, the classical four-membered beta-lactam ring, which gives a very characteristic absorption at 1765 centimeters to the -1. The final structural elucidation came from the interpretation of nuclear magnetic resonance data. Interestingly, it had a 5-membered ring like the penicillins, although sulfur was not in the ring itself, and 3 asymmetric centers. This was a structurally unique beta-lactam. It was very broad spectrum, but there was a problem.
Early in the manipulation of the thionyamycin broths, one of the earliest observations that people made was that the antibiotic, what we called self-destruct, or in the more colloquial, committed hara-kiri. That is, every time we took a culture which we attempted to purify, we knew that by spectral methods we had achieved a purification. But whenever we lyophilized the sample, the next morning when we came back, there was no antibiotic in the evaporated sample.
They had an extremely effective antibiotic, but couldn't easily isolate it in quantity. What was happening? What was there about this structure which was causing it to break down when you tried to concentrate it?
We felt from the very beginning that one of the more rational reasons for its chemical instability was due to the fact that the molecule was a highly reactive molecule and contained a very reactive functionality, namely the amino group. And so a logical hypothesis would be that the reactive amine of one molecule could interact with the reactive beta-lactam of another molecule, and the molecule would self-destruct, sort of commit an intellectual hara-kiri. This indeed was firmed up by the actual measurements of the decomposition of thynomycin than it was by molecular. Very quickly we determined that, for example, acyl derivatives of the amino function were chemically stable. However, unfortunately, they had lost the most important part of the biological profile, namely the anti-pseudomonal activity. This we could restore by incorporating another basic function in the molecule. For example, the glycyl amide of thynomycin, again, is fully active against Pseudomonas. However, it also is unstable. So we had sort of a paradox. We needed the amino group for its very excellent broad spectrum, especially anti-Pseudomonal activity, but we had to avoid it in order to create a stable compound. This we solved by converting the amine to a relatively non-nucleophilic nucleophile. That is, we delocalized the electron pair on the nitrogen by spreading it out over a larger group, and this could be realized by making either amidines or formamidines. And indeed, N,4-imidoyl thynomycin is imipenem, which is our clinical candidate.
In essence, the problem was the highly reactive nucleophilic amine group at the end of the side chain. The way around this was to distribute the charge by converting the amino group into the formamidino moiety. This enables the charge to be delocalized about 2 nitrogens while retaining the antimicrobial properties. But another problem then emerged. All the testing thus far had been carried out in bacteria grown in isolation. When the clinical candidate was tried in mammals, it was broken down by an enzyme in the kidney, reducing its effectiveness.
Intellectually, there are 2 ways in which this problem could be attacked. The first, by devising a specific enzyme inhibitor to this enzyme which destroyed thynomycin. The other would be to design a molecule which was stable to the enzyme. Our efforts to attack this problem were facilitated by the observation that the biologists made that the enzyme is localized in the kidney, and indeed they localized and identified this enzyme and identified it as a kidney enzyme. It's unusual that this molecule, thynomycin, is stable to bacterial beta-lactamases, but in reality is unstable to a mammalian beta-lactamase, which catalyzes its decomposition. So we started a screen based on the isolation of this enzyme, and by screening a few compounds which were structurally related to thynomycin, we were able to determine enzyme inhibitors, able to isolate enzyme inhibitors of very modest potency. However, by extending the structural analogy even further and making the molecule more like thynomycin, we were able to come up with an inhibitor which at equal weights would completely inhibit this enzyme. We added the cysteamine functionality of the compound, which we now call psilostatin, so that its pharmacokinetics are exactly matched to those of thynomycin.
So the clinical candidate is actually a twin drug. The modified natural product, imipenem, and a compound similar in shape to imipenem called psilostatin, which blocks the active site of the enzyme, thus enabling the antibiotic itself to circulate around the body without being decomposed too rapidly. The next stage is to make the drug. The ideal method is to exploit the ability of the natural organism called Streptomyces cattleya to make it itself by fermentation, in the way that Glaxo make the precursor for ceftazidime. But here they encountered yet another problem.
We were very disappointed to find that the microbiological titer of the fermentation had not appreciably changed in the 3 years or so of its current development. Historically, an organic chemist would hate to come up in competition to produce an antibiotic against microbes because they will lose in virtually every instance. However, in this instance, it would have not been possible for us to commercialize thinomycin because of poor fermentation yields. So there's only one very painful alternative, and that is to make the antibiotic by total synthesis. Total synthesis is not unheard of in the pharmaceutical industry, but it is a very hazardous undertaking because of necessity, because of the complexity of a molecule of this type. Any total synthesis is going to be very long. This will involve many steps and hence make a costly drug.
Imipenem is a carbapenem, typified by this bicyclic structure. The main problems in synthesis in this area are due to the high ring strain and instability of the beta-lactam ring. How did Bert Christensen approach the problem?
Basically, there are many ways of making carbapenems that are intuitively obvious to the synthetic organic chemist. And one could involve an existing methodology, but we felt that we needed some new methodology in order to make this unique ring system. Thinomycin is the most unstable of all of the commercially available antibiotics, beta-lactam antibiotics. And so we felt that any synthesis would have to involve the generation of the bicyclic system at the penultimate stage, because we wanted to avoid carrying on this reactive bicyclic system through many chemical steps. We were also faced with the problem of having to generate this unstable chemical substance under very mild conditions. So we felt if we could generate a very reactive substance under mild conditions, that we would have a chance of achieving this synthesis.
The mild reaction conditions were provided by a diazo-insertion reaction involving the generation of a carbene. Having created a diazo moiety on what will be C3, it's decomposed using rhodium acetate as a catalyst, creating a carbene. This is a highly reactive intermediate which is electron deficient, having only 6 electrons in the valence shell. It's so reactive that it inserts into the adjacent nitrogen-hydrogen bond. This powerful synthetic method leads to the bicyclic beta-lactam. The final stage in the elaboration of the ring is the introduction of the cysteal unit with creation of the double bond. So, the highly reactive carbene offers a neat solution. Interestingly, it was Merck's experience in related synthesis of antibiotics that led them to it.
We relied on some of our earlier work in making oxypenicillins, which are even more unstable than thinomycin. And here we had used a decomposition of an organic diazo compound, and this reaction involves the metal-catalyzed decomposition of the diazo compound to generate a carbene. Now, carbenes are very reactive, and we felt that this carbene would insert in the NH bond of the azetugenone. This was not a known reaction at the time, but we felt that mechanistically this reaction would prevail. And so our initial studies were just to put really 1 or 2 people on the development of this reaction.
And here it's important to note that this reaction, like every other piece of synthetic chemistry, was only made to work by developing the conditions in the lab. This diazo-insertion reaction is a very good example of the intimate relationship between the initial stages of conceptualizing what might happen in a chemical reaction and then actually working on the reaction itself. It's necessary to be able to create the conditions which will result not only in the desired product, but also the most efficient way of obtaining it. In this particular case, the products turn out to be nearly quantitative.
This is the key reaction in our carbapenem synthesis. This is the reaction that forms the bicyclic carbapenem nucleus. The reaction consists of a substrate, which is an alpha diazo beta keto ester, about 70 milligrams. It also contains the catalyst for the reaction, a rhodium catalyst. It's at 1/200th of a mole percent, or about 0.5 milligram, and the solvent, which is dichloroethane. The reaction is heated under a nitrogen atmosphere just below the boiling point of the solvent in this oil bath. The reaction at this scale and temperature is going to take about 10 minutes. This is approximately the scale that we normally run reactions of this type in the laboratory. Now, it's clear that you need to run this much larger in a factory process. And in fact, this reaction has been scaled up thousands-fold and is now used to produce the same crucial intermediate in multi-kilogram quantities.
And this is the next stage where the synthetic chemist is required, the scaling up. As Chris Newell of Glaxo observed, the reagents used in the laboratory may not be appropriate for use at full production scale. Inexpensive, safe, and high-yielding routes must be derived. For some products, a totally different synthesis might be required for commercial production. In the case of the diazo-insertion reaction, however, the commercial process still involves the same reagents and conditions, a very successful reaction. What was it that made them think of this esoteric route to ring closure?
Basically, one finds that you always return to your thesis. And my particular PhD thesis was the reaction of diazoketones. And so once the basic strategy had been laid out, it was fairly obvious, even though it's by analogy, that one way to achieve this reaction would be via carbene insertion through a generation of a diazo compound. With the establishment of the diazo insertion technology, this established the cornerstone of our total synthetic strategy. However, it was, while an important step, and probably the most important step, it was only one part of the overall strategy of designing the thionylamycin molecule. In particular, the major problem that remained was really the generation of 3 contiguous or adjacent chiral centers, and we needed to overcome this as part of the overall strategy. This was achieved by starting with a chiral building block to begin with, namely aspartic acid, which is cheap, readily available, and of course is chiral. At this point then, we decided that we would use this chiral center, which is the C5 center of imipenem or thionylamycin, and would use this center in order to direct the chirality of the existing chemistry and hence generate the chiral centers based on the chirality of the starting material.
From aspartic acid, the first step is the formation of the beta-lactam ring, retaining the desired stereochemistry. Next, the beta-lactam nitrogen is protected, and more importantly, the carboxylic acid is replaced with a functional group, which is later elaborated into the key diazo precursor. The subsequent acylation of the beta-lactam ring is stereospecific and occurs trans to what was the carboxylic acid group of aspartic acid. Finally, stereoselective reduction of the acetyl group using a hindered reducing agent completes the formation of the 3 adjacent chiral centers required for imipenem.
As a result of these efforts, we feel that we have produced probably the broadest spectrum and most potent antibiotic that's available to the clinician today. So in spite of all of the man-years of effort and the dollars involved, we feel that it was well worth development.
The results of the efforts of these 2 companies have been 2 very important antibiotics, arrived at by 2 different routes. Ceftazidime is the result of the manipulation of cephalosporin C, while imipenem has been synthesized following the discovery of antimicrobial activity of a fungus from a screen. Imipenem could arguably be the new starting point for subsequent generations of carbapenem antibiotics, which will be made by semisynthesis in a similar way to ceftazidime. It should be noted that both Glaxo and Merck have very similar research programs, so either could have come up with each of the compounds as a clinical candidate. Finally, these 2 case studies are prime examples of the fact that before anything can be synthesized, a detailed understanding of the biological framework is required. They also show that, apart from anything else, drug research has an element of risk, and the successful practitioners shorten the odds for themselves.
It's very important to have luck in research, but I think my experience tells me that the more you practice, the luckier you get.
Like all screening programs, this one relied on an element of chance, like others. Specifically, we had to screen many, probably hundreds of thousands, maybe even millions of cultures in order to achieve the isolation of a culture which exhibited all of the properties that we wanted.
I think my biggest contribution to this project was actually to say to the chaps in the lab, this is the compound you should make next. I've done that many, many times, but on this occasion I was right.
In order to be successful in drug discovery, I think one needs to be a variety of, have a variety of hats. It involves a combination of medicinal chemistry, biological chemistry, in this case microbiology, and certainly synthetic chemistry, because in order to achieve the total synthesis, for example, of a complicated structure of this type, one has to be primarily a synthetic chemist, because ultimately, no matter how good one's ideas are, you have to be able to make that compound and put it in a test tube.
Well, that's the end of the original video sequence 12 of S344. Now, I'm here with Dr. Mike Marriott, the Therapeutic Director for Microbial Diseases at Glaxo Wellcome, and we're discussing some of the advances that have taken place in antibiotic research over the last 10 years or so since that video was made. Now, Mike, in those two case studies, we've seen both the synthetic approach that we discussed earlier, and we've also seen an approach using natural products as source materials. Now, what's the current state of play at Glaxo Wellcome regarding these natural source materials?
Oh, we still regard natural products as a potential source of novel antibiotics, but we can't afford to spend the length of time that it took to work out the structure of thionomycin. We need to know as quickly as possible what the structure of the compound is. So nowadays, we've built up a library of pre-fractionated natural product samples. Each member of that library contains maybe 10 or 15 different molecules, but once we find a hit amongst that pool, it's relatively easy to fractionate it and get to the structure nowadays in a matter of a few weeks rather than months or even years in some cases.
So you're screening these in the same way, like these natural products?
Well, you've got to think in terms of 50, 100,000 molecules. We can't use the same agar screening techniques that were presented in the video. Now, we've moved on to microtiter plates. We use nutrient broth to grow the organisms in. We use 364 well microtiter plates. We use a robot that loads the plates up and reads the plates for us and puts all the data into the database. So a huge amount of automation has gone in order to speed up the process.
What's the main area of intense research these days?
In the antibacterial field, I would say it's sequencing the bacterial genome. There are something like 15 different species of bacteria whose genomes have now been sequenced almost completely. In other words, we know every single target there is for a novel antibiotic in those bacteria.
So when you say target, are we still talking about the old strategy of cell lysis as the Achilles heel of the bacterium?
We are, in the sense that we want antibacterials which kill the pathogen. So we try to sort out, and you've got to imagine or remember that a bacterium probably has something like 2,000 genes. Not all of them represent lethal targets, which is what we're looking for. So we sift through the data, again, using the computer. This information is all stored in databases, pulling out those targets which we believe are going to be lethal, and those targets which are present in a wide variety of different bacterial species, because we're looking, of course, for broad-spectrum antibacterials.
Yeah, it would seem to me that once you've actually cracked the genetic code of a bacterium, there's not a great deal that the bacterium can do. I mean, in the past, the bacterium has become resistant because it's adapted, but how on earth, does it adapt its genetic code?
Oh, yes, that's the way that the bacterium becomes resistant. Yes, it mutates, and either the target mutates and becomes different, or it acquires resistance mechanisms, DNA, if you like, from other bacteria. I would like to believe that, yes, it is the end, having all this information, but experience teaches me that bacteria are remarkably ingenious little organisms. They seem to find a way around almost anything we throw at them.
So let's just recap. We've got this vast amount of automation, we've got combinatorial chemistry, we've got genome research, we've got the use of natural resources, natural products. Why on earth do pharmaceutical companies spend so much time and effort on antibiotic research?
Because the simple fact is that there are still people dying of bacterial infections. The bacteria are very good at becoming resistant to whatever we throw at them. There are bacteria resistant. The staphylococci, for example, are resistant to all antibacterials that we have except for one. There are already organisms, pathogens, which are resistant to that one antibacterial. It's only a matter of time before the staphylococci acquire that resistance as well. In fact, there's evidence from Japan and from Chicago that that is actually starting to occur. So very soon, and this is a real fear that we feel and organizations like the WHO feel, is a nightmare scenario. We were going to have organisms around for which we have no drugs to treat them.
Do you think there will ever come a day when we do have the resources to beat bacterial infection?
Completely? Well, I mean, I wouldn't be doing my job properly if I said no. I sincerely hope that we do end up in that position, yes. We will be able to treat all bacterial infections. But I do feel that bacteria are remarkably ingenious at overcoming whatever we throw at them.
They are indeed, but I mean, if you look at the developments that have taken place over the last 10 years, which is what we've been doing today, they've really got a bit of a challenge on their hands as well, haven't they?
Oh, yes, I like to think so, too.
The research that was done in the last 10 years, if you can extrapolate that to the future, I mean, goodness knows what we'll be doing.
Oh, yeah, I'm sure if we make the video again in 10 years' time, we'll see some enormous steps forward.
Well, I look forward to seeing you back in the studio in 10 years' time to talk about that. Thanks very much for coming along today, Mike.
Thank you.
Mike Marriott, thank you.